Hello, good morning, everyone. Welcome to the MPG Primer. Just a reminder, next week there's no MPG meeting, but there is the MPG Scientific Retreat, so hopefully everybody signed up for that. And then the following week we have Mark Holbeck talking about epigenomics. And today we have Austin Argentieri. He is a postdoctoral research fellow in the Analytic and Translational Genetics Unit at MGH Hospital, Mass General Hospital, with academic appointments at Harvard Medical School and the Broad. He previously completed his PhD and postdoctoral research at the Big Data Institute at the University of Oxford. His research is focused on large-scale analysis to understand the genetic, biological, and environmental determinants of human aging and age-related diseases. And today he's going to talk about scalable proteomics in disease research. Thank you very much.
Cool. All right. Thanks, everyone, for being here earlier in the morning. Can everyone hear me okay? All right. So today I'm going to talk about scalable proteomics in disease research. And here we're really going to talk about something specifically. We're not going to talk about sort of cell-based proteomics or spatial proteomics of any of these things, but really blood-based proteomics to empower genetic association studies and studying disease at sort of biobank scale in either large biobanks or epidemiological research cohorts. Okay, so let's get a little sense of the scale of what we're talking about here. We're talking about genomics, 20,000 genes, more or less. That expands upwards if we start looking at RNA and transcripts, but then that really starts to expand and explode out when we start to talk about proteins. The current count estimate seems to be maybe we think we know maybe 100,000 different proteins, but when we start to talk about different post-translational modifications on those proteins or protein-protein interactions that lead to different types of protein variants, we really start to get into a scale of a ton of sort of protein variation, all of which can ultimately have an influence on disease-related pathology and mechanisms. As you'll see, the technology that we have so far for doing sort of large-scale proteomics is nowhere near capturing all of this variation, but it's the goal where we want to get. But ultimately, what we're talking about is a layer of biology that has many, many different aspects that we're going to try to capture in these different types of studies. I'll also just put a quick overview here of some of the applications we're thinking about when we're thinking about using proteomics in, again, these sort of biobank and epidemiological cohort study contexts. One is early detection of disease, and this is either using proteomic information to be able to detect those who already have disease, but in an early stage that's possibly undetected, but also to use especially blood-based proteins as predictive biomarkers for future disease among those who don't currently have any disease yet. So this is one of the big areas where there's been an explosion, sort of population proteomic research in the last couple of years, especially after a few large data sets have come online. Another area where we're thinking about applying large-scale proteomics is understanding the functional implications of genetic variants and genetic associations we find in different genomic studies. This is, being able to take in the same data set genetic information variants that we find associated with a certain phenotype or trait, and then look downstream to see is that genetic variation leading to a functional change in different proteins, helps us understand which genetic associations we find have functional consequences that we care about. Another area where there's been sort of a broad and ongoing application of proteomics is in drug target discovery, and for drug discovery more generally, using sort of protein biomarkers to understand who's a responder or not a responder to a certain treatment, to help us understand, again, responses to these and understand and develop these different types of treatments. And then also applying that more broadly in a sort of more personalized or precision medicine context, where we're using that information, using these protein biomarkers of disease risk, these biomarkers of disease treatment or non-response, to be able to ultimately develop more personalized treatments based on someone's response to drugs, or to understand who's at high risk and low risk and might need particular attention. All right, so here's an overview of some of the different topics we're going to go over, and I'll start first by sort of an overview of some of the major technologies we use to measure proteins, particularly in blood, but also across a number of different other tissues, sort of at scale in these different epidemiological cohorts and biobanks. These can broadly be split into two different types of detection technologies. You have affinity-based technologies, things like SomaLogic, SomaScan, and Olink, where you're using sort of a capture molecule or a detection reagent that's going to have an affinity to or bind to the specific target protein. And then we're going to use that to detect and quantify those proteins. Or you have mass spectrometry. SEER is one example where they use mass spec plus a certain nanoparticle approach, but there's lots of different platforms for mass spectrometry as well. Before I go into the specific platforms, it's worth understanding a bit about the technical challenges when we're trying to measure proteins, especially from plasma or blood. There's an enormous variation in the abundance and concentrations of different proteins in blood. Here you can see sort of an illustration of this going from albumin, all the way on the left here, all the way down to different types of signaling proteins and cytokines. You can have many orders of magnitude of difference of concentrations of these proteins in blood or in different tissues. And that leads to a real technical challenge in trying to develop the technologies to capture and measure these all within the same technology or within the same panel or plate. And this has been one of the major limiting factors in being able to scale up proteomic detection technology in terms of number of proteins that can be captured in the same technology. It's been growing steadily over the years, but you'll see that we're still on the order of being able to capture thousands, not tens of thousands or hundreds of thousands of protein variants. And that's largely due to some of these challenges in terms of abundance. Another technical issue that's specific to some of the affinity-based platforms is this idea of cross-reactivity, where we're using different detection reagents or capture molecules, like I said, whether they're antibodies or aptamers, to detect and capture these proteins, but they may have non-specific binding to other protein targets that they weren't intended for. And so different platforms have developed specific approaches to try to deal with that in different ways. So let's look at the first one. This is one of the major technologies that's used in a number of cohort studies. SomaScan, they detect proteins by using what they call somamers, which are just modified aptamers. These are oligonucleotides. They're short, single-stranded DNA sequences that they specifically modify so that they have a very specific binding affinity with whatever the protein target is. You can see an example over here of a protein, and then this is the somamer here with the unmodified sort of DNA backbone in green, and then some of the modifications they made here to that DNA sequence in purple to have the more specific binding to the sort of shape and interaction with that particular protein. The way this is then all quantified is that these somamers will then bind to the different protein targets with some reasonable amount of specificity. Those proteins will then be denatured, washed away, and then these somamers get hybridized to complementary probes on a DNA microarray. And then we use the intensity of the fluorescence signal that corresponds to these to figure out the relative abundance of the protein in the original sample. That means when we're measuring proteins from SomaScan, we're getting relative quantification, not absolute quantification. So we're not getting the sort of nanograms per milliliter, for example, of the amount of protein in blood, let's say, but we're getting the relative concentration of across all the samples for the same protein. How much does this sample have? The current platform from SomaScan can measure 11,000 proteins. It's probably one of the largest ones that we have to date. But again, that's kind of a drop in the bucket of what ultimately we might want to measure. But you can see they've sort of really been rapidly expanding the scope and scale of their platform over the last 10 years in terms of how many proteins they can measure. It's relatively high throughput, 1,000 samples a day, at least. You need 130 microliters of plasma for this one.
Can I ask a question, Austin?
Yeah.
For the total throughput of detecting proteins, does it vary depending on if you're looking at a nice tissue like blood or if you're looking at something a lot more
the tissue,
gunky?
Yeah, the throughput will depend on the tissue of interest. Pretty much everything I'm talking about, you can assume is about blood for right now, because we're then talking about scaling this up to biobank data where they're doing this on plasma samples. But a lot of these technologies can use other sample mediums, whether it's saliva or other tissue types directly. They can do it in other animals, but all of these might change depending on if you want to do that. Okay, the other major affinity-based proteomic platform where you've probably seen the largest explosion of data in the last couple of years is Olink. Olink has their own special sort of affinity-based approach to address this cross-reactivity sort of specificity problem. They use what they call the proximity extension array. And all this is they have two different paired antibodies, both that have a specific binding to the protein target of interest. You can see this here. So here we have the sort of protein analyte. We have the two different paired antibodies that bind to it. And then each of these has their own oligonucleotide sequence that's attached to it. When they both bind, then those two different short single-stranded DNA sequences hybridize together. And then this hybridized DNA then becomes the surrogate for the protein itself. We then extend that, amplify it, read it out using either qPCR or next generation sequencing. And we use that to, again, get the relative quantification or abundance of each of these proteins compared to all the other samples, not absolute. Some affinity-based proteomic platforms can read out, or at least have the possibility to read out, absolute quantification of protein expression. None of the major ones do right now. There are some smaller companies like Nomic Bio and some other companies like Alamar that are working on that now. But when we're talking about the major platforms that currently exist, and importantly, the major data sets where these data have already been generated, we're talking about relative quantification. And that leads to some challenges on the analysis side that we can talk about later. But again, high throughput, very low sample volume needed, only 1 to 2 microliters. When we're talking about these affinity-based platforms, there's a number of sources of variations that can make reading out these proteins difficult. We'll see later on this will have an impact on the reproducibility of using a number of these platforms. But this is everything from what's the detection limit in your sample of interest for the specific protein. There's different ranges of binding affinity that these different capture molecules have. Differences in how you handle your samples will change some of the expression readouts. You can also have genetic influences that might change sort of different amino acid sequences that can change the 3D structure, or some post-translational modifications that can also influence the way these molecules are going to be binding to your target protein. So all of these are possibilities for variation to creep into what you're doing. And it's important to sort of remember this, especially on the downstream analysis. The other main area the other main technology that's being used at the moment for protein quantification is mass spectrometry. It's not affinity-based. You can see sort of a nice sort of figure of the whole workflow over here on the right, but essentially you take proteins, you break them down into peptides, you separate them according to their mass and their charge, and you ionize them. And there's different methods that are available for separating them out. The most popular or well-known is maybe liquid chromatography. And there's different methods for ionizing them as well, like electrospray ionization. But in any case, there's a lot of different platforms and different machinery for doing this, but this is the general idea of what we're doing. It's much lower throughput, but you get a lot more variability on what you can read out. So you can detect peptides, you can detect whole proteins, you can also detect certain types of post-translational modifications, and different platforms might have different abilities for what PTMs they can actually read out. But there is the possibility to get more fine-grained information if you're interested in more sort of protein variation or information on the peptide level. It again is often relative quantification, but here also absolute is possible, and it's just going to depend on the specific platform you're using. So here's just at a glance what we've talked about so far. These are the platforms, how many proteins we're getting at. Overall, we're really still a drop in the bucket for how many proteins we can measure based on how many proteins we know are out there, and certainly a drop in the bucket on protein variants. But this is where we are right now, and you'll see in the next couple sections we're still able to do, even with this amount of information so far, a lot of work and a lot of reliable work and robust work in terms of disease prediction and understanding other important traits and phenotypes we want to study at a population scale. So.
Yeah, I have a quick question. For the protein that you're quantifying, are those non-protein, or are you doing like de novo, finding as well? Because I imagine the
Yeah.
probe are designed for non-proteins.
Exactly. Yeah, that's a really good point. Especially the affinity-based platforms, they're all targeted panels. So they have made decisions based on choosing these specific 5,000 or 11,000 proteins, for example. They usually do it by trying to get as broad a coverage they can across different sort of biological pathways and different proteins where there's some evidence they might be important for disease prediction or drug development or things like that, but it's all biased and completely targeted and selected. Mass spec, they say, is more unbiased, but some of them still do introduce some amount of selection into the process in different stages, especially some of the ones like SEER proteomics that includes like nanoparticles plus mass spec. They are, making decisions there. But even if it's, quote unquote, more unbiased, we're still only getting a readout of, proteins on the order of thousands. So we're not getting like unbiased readout of all 100,000 proteins or something like that.
I had a related question to that in terms of just genetic variation. How much will that affect your affinity? Or do they design different aptamers for maybe different protein mutations?
Right, like are there different aptamers or antibodies for different proteoforms that are a result of different genetic variation? It's a good question. I don't know 100%. On the Olink platforms, no. On SomaScan, I think they have some aptamers and they can capture a few different proteoforms for some proteins. But whether it's as intelligent as you're describing, where they're like specific ones for known variants, it's unclear. But we'll see in some of the later slides, there's a really broad spread across even the proteins we can measure on these platforms on how much expression will be influenced by genetic variation, everything from like 1% explained to like up to 70% explained. So it also just really depends on the protein. And that is kind of the takeaway all the way through for a lot of this, is that the reliability of all this and the associations is going to be dependent on like protein by protein. Okay. So that's how we make the data, or that's how these data are being generated so far in population cohorts. And again, that gives you a sample of the 3 major platforms that are out there. So where can you actually access these data and start using it for your genetic association studies or studying specific diseases or traits? Here's just a non-exhaustive overview of some of the major sources of data that you can look at right now if you want to study sort of proteomics at scale, meaning like at the sort of larger scale of what exists currently. UK Biobank, the proteomic data came on the scene a few years ago in 50,000 participants, which was far and away the largest data set that had been produced so far. On the left here, you can see a bunch of other data sets, that are all international. So we have the FinGen cohort, which is, of course, a lot of the FinGen team is also here at the Broad. They've got 5,000 participants so far, but a roadmap to 10 to 12,000 in the next few years. Scalable is a consortium of a number of different cohorts that have paired genetic and Olink data. Across those cohorts, you have 70,000 participants. China Kadoorie Biobank and Fenlon is another study in Europe. But then in the US, we also have a lot of different cohorts from the TopMed program. If you're not familiar with TopMed, it's a program to do sort of standardized multi-omic assays across a number of NIH-funded cohort studies in the United States, where they're doing sort of genotyping plus proteomics plus other omics like methylation and metabolomics as well. So we've got proteomics online now, sort of that are done in a standardized, harmonizable way across these cohorts for a number of different data sets. I think this is what's out there as of the last month or so. We've got 30,000 samples with proteomics data, either Olink or SomaScan across all these US cohorts. So there is a lot of proteomic data out there. The sort of question is, to what extent can we bring it all together and harmonize and pool it? I think that will depend, but we can talk about that in further slides. There's also way more coming. So the UK Biobank announced last month that they're doing 600,000 proteomes. So the full 500,000 person cohort will all have proteomics done on the new Olink 5,000 protein platform. And then for 100,000 participants, they're going to do a repeat sample as well. So the amount of data has exploded in the last few years, and in the coming few years will explode even further to just a scale and size that's just been, unlike what we've ever seen before. So stay tuned for those data. I think they say in a few years from now. Okay, I'm going to now go into some of the studies that are out there in terms of what we've learned over the last few years from a lot of these population-based studies of protein expression. I'll go through a couple examples. I'm not going to necessarily go super into detail on any of these studies, but it's more meant to give you an idea of the kind of universe of what's out there that people have been doing so far with these kinds of data. And I have all the papers and references up there that you can go into, further detail. Feel free to ask questions on any of these as well, although with the caveat, I'm not an author on most of them, so I'll try to answer whatever I can. But if we first talk about studies on genetic regulation of protein expression, this is mostly done through what's called pQTL mapping. A pQTL is a protein quantitative trait locus. It's much like an expression QTL or methylation QTL. All we're doing is we're looking at the effect of a specific genetic variant on protein expression using protein expression data from these different platforms, mostly so far Olink, mass spec, and SomaScan. And then we're mapping those into what are the cis pQTLs, like within 1 megabase, usually of the gene that codes for that specific protein, or what are the trans pQTLs that are sort of beyond 1 megabase away from the gene that codes for the specific protein that we are looking at. There's been a lot of these studies across, but the idea is to get an understanding of the sort of short range and long range or indirect sort of genetic regulation on these different proteins that are circulating in blood. So a couple of years ago, after the UK Biobank data came online, there was this large study in Nature that was looked sort of comprehensively across all roughly 3,000 proteins they had there, where they had paired genotyping data and whole genome sequencing data. They found a ton of pQTL associations. It's far too much to go into detail here, but about 81% of them were previously undescribed, just because the sheer sample size of this was just far greater than anything that had been done before. But they demonstrate that when pairing the genetic information with the protein expression data can be really powerful through using causal techniques like Mendelian randomization to be able to do more queries for possible drug discovery, understanding potential causal influences of proteins on diseases of interest. One example they give in the paper is recapitulating sort of known relationships between PCSK9 on cholesterol, stroke, heart disease, other related traits like that. So this was really kind of a flagship paper for demonstrating that we can do really large scale sort of genetic regulation mapping of these protein expression data at scale, and could be a possibly really valuable tool for further drug discovery, discovering sort of novel potential causal relationships between proteins and different traits that we care about for different diseases and different treatments. There's also been more recently some really cool studies that have come out looking at pQTL mapping in child and adolescent participants as well. This came out within the last month, I think. This was using mass spec-based proteomics, but again, they did plasma proteomes of a little bit more than 3,000 individuals, 5 to 20 years old. But one of the interesting things they show here is that there's really interesting, and I'm showing some of them down here below, there's really interesting sort of protein abundance trajectories of a number of proteins across this sort of more developmental period of life. These include sex hormone-related differences for proteins, but also bone development proteins, for example. But they also show that for 63% of the proteins that they studied, the pQTL, so the genetic regulation for that protein, contributes more to explaining the variance in expression than age and sex and BMI combined. So at least at certain stages of life, for a lot of proteins that are being studied, actually genetics is having a large amount of effect on explaining variance in these different phenotypes. And they did, because they have genotype information, they did more causal association mapping. You'll see that's a trend across all of these different studies. This paper came out just a couple of days ago, but there's also been a lot of effort in the sort of rare variant space to look at using whole genome sequencing data in the UK Biobank to sort of map associations between rare and non-coding variants and protein expression. Again, this example is 3,000 proteins in 50,000 UK Biobank participants, but they find a lot of associations. And one interesting thing they end up finding from this is that unlike some of the protein coding variation, rare non-coding genetic variation seems to be equally likely to be related to either increased or decreased protein levels. So we're learning a whole lot about the genetic regulation of these proteins in the last years, or even the last few months, to be honest. That is going to be really helpful as we continue to go forward, trying to understand mechanisms related to all these traits and phenotypes we care about. So the next major area where there's been a ton of activity in the last couple of years is disease prediction. I'll give a couple, key examples here. This was a great paper that came out a year ago or last summer from Claudia Langenberg's lab. This was in the UK Biobank. They looked across 218 common and rare diseases. And this is the real value of doing this at biobank scale, where you have linked health records, is you can kind of look systematically across tons of diseases and traits. So they're looking at the relationship between plasma proteomic expression at baseline among people who don't have disease, and whether it predicts development of disease or disease diagnosis, up to 10 years in the future. One of the really cool things they find in this is that, yes, they studied 3,000 different proteins, but you can find predictive models that use just as few as 5 to 20 proteins that can predict, future risk of these diseases more accurately than sort of basic clinical demographic information for 67 different diseases. But also they can outperform models that are sort of clinical information plus clinical assay data, things like glucose, HbA1c for diabetes, things like this. And so the takeaway from this is that with just a handful of proteins and information on just 5 to 20 proteins, we have the possibility to be able to, in some cases, predict future risk of disease more accurately than even the sort of gold standard clinical prediction models that we have now. So there's a lot of activity right now thinking about, well, yes, on the one hand, scaling up these platforms to do many more thousands of proteins that we have, but there's also a lot of activity, especially after seeing results like this, thinking about how can we also scale down these platforms to have, say, panels that can be cheap, low cost, measure maybe 20 proteins, but provide better sort of clinical prediction accuracy than the sort of gold standard clinical information that we use now. This was a great paper that sort of demonstrated the possibility of being able to do something like that. There have also been papers that have come out on cancer prediction. This was one from a research group at Oxford over in the UK. They find lots of associations between, again, in the UK Biobank, between proteins and incidence of different cancers. Lots of them are relationships where the cancer is diagnosed more than 7 years after the protein's measured. So, again, in some cases, able to identify, future risk of cancer many years out when you don't currently have it, or at least you don't currently have a diagnosis of it, but the biology has probably still been accumulating for some while. And in any case, I won't go into any of the sort of specific details here, but again, we're getting these really comprehensive maps across some of these different studies of the relationship between a lot of these different proteins and different outcomes, sort of culminating in some work like this. This came out in Cell maybe a few months ago, where looking in the UK Biobank, they just looked comprehensively 3,000 proteins to almost 2,000 different traits and diseases. And they developed this really sort of great phenome-proteome map. I think the URL is in here, proteomephonemeatlas.com. They also pulled in all the genetic information. So for each of these protein to trait associations, they then do Mendelian randomization. They figure out, try to at least suggest which are the ones that have possible causal relationships between them. So in any case, there's been a ton of really comprehensive mapping that's been done sort of systematically across the data we have access to in some of these big cohorts. That's just a treasure trove of information to start looking through. I think these days, if the protein is on some of these Olink or SomaScan panels, you have a lot of information. You can go start to look up what are the diseases and traits that might be associated with this. And I can tell you that a lot of the pharma companies are mining this right now to develop new programs for therapies and drug targets as well. Okay, I'm going to touch briefly on another area that I think is cool, but I'm biased because I do aging research. But there's been a lot of interesting studies coming out in the last 5 or so years that have been trying to study how proteins change with age and how they can teach us a little bit about the biology of aging and provide some tools for prediction of age-related diseases. This was a study that our group led. We developed a sort of proteomic aging signature in the UK Biobank, the idea being that a lot of age-related diseases and age-related traits share common aging biology. And if you can capture that in blood-based proteomic information, then can that start to be a signature or a tool that gives you powerful prediction of many different age-related diseases? That's certainly what we found here. We can find we developed this proteomic aging signature that predicts future risk of mortality in 18 different chronic diseases, and also lots of different aging-related traits like cognitive function, frailty, physical decline, these things. But then because there's all of these data sets that are online now, we can start to validate these models in other data sets. So we validated in FinGen proteomic data and proteomic data from the China Kadoorie Biobank as well. There have also been some other pretty cool papers that have come out in the last year. This is a paper in Nature from December 2023, not in the UK Biobank, but in a few other data sets that had SomaScan data, where they were taking information on which proteins might be more sort of organ or tissue specific. Here they use GTEx, but there are other databases you can use for that as well. But then starting from the premise that as these organs and tissues start to degrade, they're going to start to leak proteins back into the bloodstream. And so you can actually still measure some proteins that should be more organ specific using just plasma-based proteomic samples. They then develop these sort of organ-specific signatures of aging for a bunch of different tissue types and organs, and that these show pretty cool sort of specific associations to traits and phenotypes and diseases related to those. So a good example is, this muscle protein aging signature is associated pretty specifically with just gait impairment. Heart aging is associated, at least from proteomic information, is associated pretty specifically to heart attack, atrial fibrillation, and hypertension. So there's been a lot of cool work, I think, in the last couple of years on can we use actually just blood-based proteomic information to get insights about sort of organ-specific phenotypes, and how does that help us in prediction of different organ-specific diseases, but also give us a more nuanced sort of picture of the sort of overall health that's happening in a person on a more sort of fine-grained level than just overall in the entire organism. There's also been this was a paper a few years ago also in some SomaScan data showing that they could cluster different proteins in plasma into pretty distinct trajectories of changes in expression across age from 20 to 100 years old. So there's been a lot of, I think, interesting research showing that all these proteins that we can pull from blood have pretty interesting dynamics across life, and we can sort of capture that information to tell us a lot about aging biology and risk of different aging-related diseases. Okay, so the next question that I want to sort of touch on here is a really important one, actually. We have all of these different platforms for measuring proteins. A lot of them measure the same proteins, not all of them, but there's a core set of maybe 2,000 proteins that across these different platforms will be shared, or they'll have sort of a similar assay for. The question is, across all these platforms, are we measuring the same protein in each platform, and can we make them interoperable? And there's been a lot of research, and there's a lot more research going on now trying to test this in real world data, where we have a number of data sets with a few thousand people that have both Olink and SomaScan, for example, or Olink, SomaScan, and mass spec. You see 2 published papers here on the left and 2 more recent preprints on this. And the answer is they're really not always measuring the same thing. These are 3 different studies where they have both Olink and SomaScan for the same participants. This is just a distribution of the correlation coefficient between the same protein across the 2 platforms. You can see there's some proteins, on all of them. These are 3 different separate data sets as well, but they sort of recapitulate a similar takeaway, which is that there is this sort of bump here on the right of proteins that are fairly strongly correlated, where the same protein on Olink SomaScan is strongly correlated with each other. But there's this very worrying huge node all the way on the left here across all of them, where you have the same protein on Olink and SomaScan, basically not very well correlated at all. So the fundamental question from this is, are these platforms measuring the same thing? Are they actually measuring the same protein? They have different ways that they detect the proteins. And the question is that difference in the way that they are detecting the proteins, whether it's from their aptamers or from the antibodies or from the mass spec, are they actually capturing different isoforms, different proteoforms? Are we not actually capturing the underlying same actual protein or protein variant across these things? The correlation results I think so far, and these are, by the way, on the left here, these are results from participants in China. Here in the middle, I believe this is from participants from the Finland study in Europe. And then on the right here is from the ERIC cohort, which is in the US. So I think the answer, sort of getting to what I was saying before, is that our confidence in these proteins across platform is really protein specific. And it's going to vary protein to protein. And unsurprisingly, this leads to changes in the genetic associations we get for the same protein across platforms as well. So if you look here on the right, for example, this is looking at the pQTL, so the beta regression coefficient for the pQTL for the same protein on the sort of bottom on the X on SomaScan and on Olink on the Y-axis. And they're somewhat correlated, but for the same protein across the two platforms, you can see that the either magnitude or just the direction of the genetic effect can be sort of widely divergent between the two platforms. So there are some when it's really nicely correlated, and there's some where it's widely divergent. So again, if we're seeing an underlying difference in probably what they're measuring in terms of the actual correlation between the expression, it's leading to differences in the genetic signals that we can map to expression of these proteins across platforms as well. Here on the left is just showing, again, in this China population, when you look at Olink proteins versus SomaScan proteins, in the middle is a core set of overlapping proteins where they find pQTLs for both of them. On the left, there's 340 almost, where they only find a pQTL for Olink, and on the right, they only find a pQTL for SomaScan. So the results are tending to diverge for the genetic association studies as well, and also for the phenotype associations here. So again, this is just looking at Olink versus SomaScan associations with ischemic heart disease in Chinese participants, some good correlation for some proteins, some opposite direction of effect, and then across Olink, SomaScan, and a few different mass spec platforms here on the right, across a number of fundamental phenotypes, BMI, age, sex, hematocrit, smoking, things like this. The number of proteins that are associated with each of these across the platforms can vary wildly. It is different that these different platforms capture a differing number of proteins, but in my mind, this is really one of the fundamental things that we are trying to grapple with as a field now, is if these different platforms are measuring different underlying sort of biological targets, which is the right one, which is the one that you want to look at in your study. So this is all to say that there is some thought that should go into which is the right platform for you. Some platforms will just be the only one that have the protein that you're interested in, and that's fine. But where there is sort of coverage of your protein target of interest across multiple platforms, there's now a lot of information like this that you can use either from the genetic associations or the phenotype association mapping, where you can start to look at what might be the most appropriate platform for me. But there's a lot of work going into figuring this kind of stuff out now.
Have people done a meta-analysis of this to compare, at least across studies, that were always the same proteins are always have this correlation, that maybe we could make models so that regardless of what data you have, you could just project it onto whatever technology, like if it's consistently biased in the same way across cohorts?
You mean in terms of the protein, like with the protein-protein correlation, or like the protein to phenotype association?
I was thinking protein to protein, just the easiest level to work with.
Just to like project onto data you have, like this is a reliable protein.
Yeah, like are they those 3 studies where you had those bimodal Pearson correlation or correlation distributions? Is it always the same proteins that are in the second hump, and always the same proteins on the first hump?
Interesting. Yeah, I don't. It's actually a really good idea. I don't know of a meta-analysis across this, but the way that a lot of this is working right now is that everyone's kind of working siloed in their own data sets, which is either because these are all in protected research environments that you can't pool, but there's also statistical difficulties with pooling sort of relative quantification data as well. But meta-analysis, I haven't seen any on this. Would be really interesting to see though. Yeah.
Sort of along those lines, was there anything done in any of these studies where they looked at the proteins that didn't correlate well? And is there something about them? Like, were they all signaling proteins? Were they all, like at the beginning, you showed different sort of classes of proteins. Like, were they the same, like something about those proteins?
The slide I showed way back at the end of the section on what platforms are out there, where there are different sources of variability. That was from, I think, this study in the middle, where they start to look at associations with lots of different sort of genetic factors or demographic factors or things like that to understand what are the sources of variation in the proteins. And some of that list they came up with were things that they could detect in their study that seemed to be driving the variability in protein expression and was seeming to explain more about like which proteins seem to be less well correlated across platforms. On the right here, you can also see one of the things that this sometimes maps onto is each of these platforms will have a limit of detection. So like Olink, for example, has a limit of detection, and sometimes a protein will be below the limit of detection. They'll still report to you the value anyway, but flag it that it was below. And you can actually see here that when you have, the number of proteins that are in these different humps can kind of be mapped a little bit to, how many of them are above the limit of detection. So like here, the ones where they're sort of all hovering around 0, there's a ton of proteins here where, less than 20% of them were, less than 20% of samples were above the limit of detection. So some of this like technical variability, I think, is what's creeping into things as well. Okay. So this leads to one of the last topics to describe, which is reproducibility across these. We've sort of gotten at the sort of across platform interoperability or like reliability, but if we talk about within platform reproducibility, one of the ways that this is measured over and over again is through something called a coefficient of variation, or a CV. The way that we do this is you have technical replicates within some of these studies where they take the same sample and they run it multiple times on the same machine. The idea is that if it's a reproducible and reliable assay, you should get the same value every time. So we can calculate this coefficient of variation, this CV, as like the percent of variability in your assay. That number should be as close to 0 as possible. All of these manufacturers will publish what is their own internal studies, official CVs. They all say that they have an average CV below 10%. You generally want something to be below 10-15% to be reliable. But these studies, these figures are from real world data, and you can see that it's actually like highly variable across platforms and sort of protein by protein basis. So if we look at Olink and SomaScan, for example, they have the previous 3,000 protein Olink and the 5,000 protein Olink here, and the previous 7,000 SomaScan and 11,000 SomaScan. One of the things they conclude in this paper is that increasing the number of proteins on the new Olink panel has led to sort of less reproducible assays. They had to make sacrifices in this sort of cross-reactivity problem, figuring out the different abundances, and it's led to less reproducibility, at least in this one study so far. SomaScan seems to be pretty low here, consistently an average below 10%. But across all of them, you see variability across the whole spectrum from 0 to 100. So in my mind, this is a problem that's not talked about enough, but the reproducibility of all of these platforms is not really just on like the platform level. Like this platform, the average CV is less than 10%, but it's really like some proteins are going to be way more reliable than others. On any of these platforms, a lot of this information is now public and out there, either in preprint or published papers. So this kind of information is also important to think about as you're thinking about choosing what platform, as you're thinking about also interpreting the results that you have in your data set. You're looking at the UK Biobank, how reliable is my association between expression of this protein and this phenotype or the genetic association or whatever. You can start to look at this information and understand, well, how much confidence do I have in the reproducibility of that expression readout? Okay, last slide. I think we're right on time. There are a couple, just sort of wrap this up and sort of digesting all this information, there are a couple considerations to think about when we want to actually analyze these kinds of data. The first should be a reassuring point, which is that protein expression is, at the moment, much more simple to analyze than genetic information. I mean, it's just a quantitative trait like anything else. So you have expression of a protein, you put it in a model like any other quantitative variable, or numeric variable. There might be considerations about Do you need to make sure you've done quality control, normalization, all these kinds of things? But it's easy to analyze, but there is opportunity for new methods and tools. There's a lot of ongoing research right now in understanding variability in proteins and what we should need to be adjusting for and all these kinds of things. But nonetheless, a consideration, like I've sort of alluded to, is if you have specific proteins that you're interested in, they might only be represented on one platform. That might constrain your choice. If you want to look at post-translational modifications or proteoforms, then those are interest of you. You're probably going to want to go with whatever mass spec data is out there. There is mass spec data out in a number of cohorts now, so it's worth looking for. The other point is that pooled analysis and meta-analysis is possible, but with caveats. In some of the work that we've been doing, we've been successfully able to build models, for example, in the UK Biobank and validate them in external data, like the FinGen cohort or the China Cadore Biobank, where they've done proteomics on the same platform. But it does require a lot of specific sort of renormalization, again, because all of these are done on relative quantification scales. So across all of these, for example, expression on an Olink platform is just on a completely arbitrary log scale. So that means that the range of your protein in one cohort might be between 0 to 100, and another one might be between 0 and 5, right? So you need to make sure that you're renormalizing and sort of developing the statistical tools to be able to make those data interoperable between different data sets. It used to be that companies like Olink recommended that you don't do that at all, and that the only time they recommended you do that is if you had a bridging sample where you had a sample that was measured across all the batches, and you could sort of renormalize them all that way. I think there are some techniques that we and other groups have developed that allow you to figure this out statistically and renormalize at least on population scale, but how well it holds up on individual scale is yet to be seen. So some early success, but we need a lot more work from the scientific community to figure more of this out. I guess the last point. And as we go forward, I think people are still figuring a lot out on what's the consensus on how we should be analyzing all these things. What are the major sources of variation? What do we need to control for? What should we be accounting for, and how we do all this? So we're in the early days. There's been an explosion of data, and everyone's trying to keep up now in terms of mapping all the associations, mapping all the major sources of confounding and variation. And it's worth keeping updated on everything that's happening because it's happening fast. The very last slide and the last thing that I'll say is if you want a great resource for diving deeper into any particular protein of interest, Human Protein Atlas is probably one of the best places to go. There's not a lot of correlation between gene expression and protein expression for a lot of proteins. So if you want to get at, for example, information about tissue specificity, probably much better to go to the Human Protein Atlas, where they've measured tissue specific expression in proteins than to go to something like GTEx, where it's going to be based on RNA, and you're going to be making a lot of assumptions about whether the RNA tissue specificity is held up for the protein as well. So they have tons of information here. They have information about tissues of interest, gene information, and have information about RNA expression, single cell information, information about relationships to cancer and other diseases. So if there was a one-stop shop to figure out more about a protein, it might be the Human Protein Atlas, and they're always updating this and expanding this. So yeah, I'll leave it there. If there are more questions, happy to take more. Yeah.
I have a little bit of a philosophical question, maybe. What's your vision on how many actually proteins are in human plasma? So we're talking about biobanks, so we're always dealing with plasma, and we see SomaScan 11,000. We see older mass spec data 600. What is the reality estimate in your vision?
The number that people kind of throw around from time to time is maybe 100,000, but in blood specifically, like what can we measure reliably, and what's there and what's not just tissue specific and not showing up in the blood? I don't have a number that I know. I don't know that anybody knows that number. I think, but it's certainly more than what we have now on the panels. But the question of whether we're going to hit a rate limiting step of until these companies can figure out this abundance problem and how to put a ton of proteins together, just have crazy different abundances. I think that's the rate limiting step right now. But I don't know.
I
I know it's
might,
disappointing, but I don't have a specific number that I know of.
I myself think about it a lot. And on the other hand, it feels a little bit like we know that antibodies can be very inspecific sometimes. And especially with the genetic variation and everything that you just talked about, and how many false positives are there actually. Maybe think we measure much more than there is actually protein in the plasma.
Right.
A bit scary for research, right?
Yeah, I mean, all of the platforms will have different statistics they will market to you on the specificity of their antibodies and all that kinds of stuff, but cross-reactivity is real and is a problem that they're all trying to deal with. There are new technologies that claim to be able to sidestep that altogether, but we'll still be influenced by other things like different genetics, different variants will create different amino acids, will create different shapes. So I don't know. It's the takeaway is possibly that like we're figuring it all out now, and I don't know that anyone has the perfect answer. Yeah.
Great, and let's thank our speaker. Well, that is time. I'll see you guys all next door at the MPG seminar. Thank you. Thank you, Austin.